Hello, my name's Robert O'Flynn. I currently work for the Yunnan Key Laboratory for Paleobiology, and I'm here today to talk to you about phylogeny, which is basically the evolutionary history of life. Now, much of the power of the theory of evolution is its ability to provide a sensible framework for understanding the diversity of life. Observations from throughout biology cannot be understood except because of evolution. As vertebrates have evolved, the same bones have sometimes been put to different uses, yet the bones are still recognizable, their presence betraying their evolutionary past. For example, the forelimb of vertebrates are all homologous structures with different appearances and functions that all derived from the same body part in a common ancestor. You can see in Figure 1 how the bones of the forelimb have been modified in different ways for different mammals. Why should these quite different structures be composed of the same bones? A single upper forearm bone, the humerus, and a pair of lower forearm bones, radius and ulna. Several different carpals and 4 or 5 digits. If evolution had not occurred, this would indeed be a riddle. But when we consider that all these animals are descended from a common ancestor, it's easy to understand that natural selection has modified the same initial starting blocks to serve vastly different purposes. Some of the strongest anatomical evidence supporting evolution comes from comparisons of how organisms develop. Embryos of different types of vertebrates, for example, often are very similar early on, but become more different as they develop. Early in their development, vertebrate embryos possess pharyngeal pouches, which develop into different structures. In humans, for example, they become various glands and ducts. In fish, they turn into gill slits. At a later stage, every human embryo has a long tail, the vestige of which we carry to adulthood as the coccyx at the end of our spine. Human fetuses even possess a fine fur called lanugo during the 5th month of development. Because natural selection can only work on the variation present in population, it should not be surprising that some organisms do not appear perfectly adapted to their environments. For example, most animals with long necks have many neck vertebrae for enhanced flexibility. Plesiosaurs had as many as 76. By contrast, almost all mammals have only 7 neck vertebrae, even the giraffe. In the absence of variation in vertebrate number, selection led to an evolutionary increase in vertebrate size to produce the long neck of the giraffe. An excellent example of an imperfect design is the eye of vertebrate animals, in which the photoreceptors face backward toward the wall of the eye. As a result, the nerve fibers extend not backward toward the brain, but forward into the eye chamber, where they slightly obstruct light. Moreover, these fibers bundle together to form the optic nerve, which exists through a hole back of the eye, creating a blind spot. By contrast, the eyes of mollusks are more optimally designed. The photoreceptors face forward and the nerve fibers exit at the back, neither obstructing light nor creating a blind spot. Such examples illustrate that natural selection is not like an engineer who can design and build the best possible structure for a given task. Workable but imperfect structures, such as the vertebrate eye, are an expected outcome of evolution by natural selection. Organisms possess vestigial structures that have no apparent function but resemble structures their ancestors possessed. Humans, for example, possess a complete set of muscles for wiggling their ears, just like many other mammals do. Whales, for example, possess pelvic bones, as other mammals' skeletons do, even though such bones serve no function in the whale. The human appendix is vestigial, i.e. it has no current function. It represents the degenerate terminal part of the cecum. In other mammals, such as mice, the cecum is the largest part of the large intestine and functions in storage, literally of bulk cellulose in herbivores. Vestigial traits can also be seen in the genomes of many organisms. For example, the ice fish is a transparent fish that lives in the frigid waters of the Antarctic. The ice fish's transparency results not only from a lack of pigment in its body structures, but also from the invisibility of its blood. Our blood is red due to the presence of red blood cells, which contain hemoglobin, the molecule that transports oxygen from the lungs to the tissues. However, oxygen concentration in water increases as temperature decreases. The water of the Antarctic, which are about 0 degrees Celsius, contains so much oxygen that the fish do not need special molecules to carry oxygen. The result is that these fish do not have hemoglobin, and consequently their blood is colorless. Nonetheless, when the DNA of ice fish was examined, scientists discovered that they have the same gene that produces hemoglobin as that found in other vertebrates. However, the ice fish hemoglobin gene has a variety of mutations that render it non-functional, and thus the ice fish does not produce hemoglobin. The presence of this inoperative version of the hemoglobin gene in ice fish means that its ancestors had hemoglobin. However, once the ice fish's progenitors occupied the cold waters of the Antarctic and lost the need for hemoglobin, mutations that would be harmful and thus would be filtered out by natural selection in other species were able to persist in the population. Just by chance, some of these mutations increased in frequency in the population through time, eventually becoming established in all individuals and knocking out the fish's ability to produce hemoglobin. All these anatomical lines of evidence, homology, development, and the imperfect and vestigial structures are readily understandable because of descent with modification, that is, evolution. Comparisons of the anatomy of different living animals often reveal evidence of shared ancestry. In cases of homology, the same organ has evolved to carry out different functions. In other cases, an organ is still present, usually in diminished form, even though it's lost its function altogether, such as the organ or structure is termed vestigial. Thank you for listening. We look forward to seeing you next time, where we shall continue our exploration of phylogeny. Thank you very much.